In this video, we will be covering the cytoskeleton and what makes up the components of the cytoskeleton. Here we have a very beautiful picture of a cell, and we can see the different components of the cytoskeleton. We have the nucleus, and we have some strings that kind of radiate away from the nucleus. And so we are going to explain and explore how each component of the cytoskeleton, which are only three, how each one is made and how they operate and what they do for the cell and for your body. Like I said before, there are only three cytoskeletons, those being the intermediate filaments, the microtubules, and the actin filaments, which are also called microfilaments. These are called microfilaments. If we were to rank the sizes from thickest to thinnest, then we would have this structure. We would have this hierarchy. The microtubules are the largest cytoskeleton components. The diameter for the microtubule would have to be 25 nanometers. 25 nanometers. The next biggest cytoskeleton filament would have to be the intermediate filament, and the intermediate filament is only about 10 nanometers. Now, the thinnest and the smallest cytoskeleton filament would have to be the microfilaments, which are also called actin filaments, and these are about 5 to 9 nanometers. The intermediate filaments, when they are created, they're created by two strands, and these strands will eventually come in contact with each other and they will start to twist. And since they twist, they become fibrous. And so we can say that the intermediate filaments are fibrous. These are fibrous proteins, meaning that they're like strands. Like if you ever gotten some string cheese and you kind of peel it apart, those little peeling parts, those are the fibrous proteins. And if you get those little strands and you twist them together, they become an intermediate filament. And since strand A is different from strand B, we call these heterogeneous fibrous proteins. So these are heterogeneous. Heterogeneous. Gene-ES. Okay. Now the microtubules are a bit different. The microtubules are made of a dimer. So this is a dimer. A dimer means two. And what it is made from is made from alpha subunits and also beta subunits. Specifically, by subunit I mean the tubulin. So we have the actin tubulin combining with the beta tubulin. And here you can see the alpha and beta tubulins stacking on each other. The actin filament is just made of one thing, and that thing is called actin. So this is just actin. So actin combines with itself and it twists to form the filament. So this one is made from two different filaments, proteins, and they twist on each other. This is made from the alpha and beta tubulins that stack on top of one another. This is the largest, this is the medium size, and this is the smallest. We will now be talking about the intermediate filaments. So the intermediate filaments, which I'll call IF, the intermediate filaments, their job is to create stable networks that withstand mechanical stress. Okay, so here we have the cytoplasmic intermediate filaments. So these are often connected to the plasma membrane. So they're connected to the plasma membrane. So that's connected to the plasma membrane. And they form cell-to-cell junctions, right? So they're going to go between these cells, and they're going to be creating these connections between the cells, right? So here we have the intermediate filaments in red. So cell A is going to be sharing it with cell B. And these are forming connections, and they form connections at the junctions, which are called desmosomes. So these connections that you see here, these are called desmosomes. And that offers cell security and cell support. So if the cell is being in contact, let's say that we have some sort of outside force, if I press down on this cell, it's not going to crumple up and be destroyed. And it's not going to be destroyed because it has these supporting connections with the neighboring cells. And these connections are formed at the desmosomes. So this offers cell support. So here we have the desmosome, and it is connecting the two cells. It's not really a good picture, you can't really see what's going on, so I showed you here on this separate picture. And here inside the nucleus, we have more intermediate filaments. And these intermediate filaments, they are called nuclear. These are called nuclear, let's write that, nuclear intermediate filaments. And their job is to support the nucleus, right? So these are going to form lamins. These make nuclear lamins that create a mesh that supports the nuclear envelope. So this kind of offers shape and support for the nucleus, right? If you didn't have this, the nucleus would just be kind of like a jelly inside its eggshell, right? So here we have the nucleus, and then here we're going to have the genetic material right here. We don't want that genetic material to be sloshing around inside the nucleus, right? Because if it sloshes around, it could be damaged, it could get destroyed or whatever. And so really, I'm going to add these nuclear lamella to the sides of this nucleus. And the nuclear lamella is going to make sure that this red thing, the genetic material, is supported and doesn't move around. Because if it moves around, it could get damaged or it could escape. And so we have these lamins in green made of intermediate filaments that offer support for the genetic material. And here we have the very nice mesh network. So this is the network of intermediate filaments that is found inside the nuclear envelope. So this is made from the nuclear lamins. Intermediate filaments, they're assembled by a variety of proteins within different cell types. And of course, we have them in the nucleus. So depending on where you find the intermediate filament, it will have different responsibilities. So for instance, all the intermediate filaments can be found within the cytoplasm. But where? Well, if we look inside the epithelial cells and we look at the cytoplasm, we can see that the filaments are called keratin filaments. And these, for the epithelial cells, this offers support. Right? Support and strength. If we look at the connective tissues and the muscle cells and also the glial cells, these intermediate filaments are called vitamin and viminin related filaments. And so that offers, it allows some cells to operate, right? So in muscle cells, if you didn't have these intermediate filaments, well, the muscle cells wouldn't operate. They wouldn't be able to fully function. Yes, they will still have microtubules, but the muscle cells wouldn't be able to function. Okay, so these are very crucial within the operation of the cell and body. Now, if you have nerve cells and you look inside the cytoplasm of the nerve cells, we have the neural filaments, and the neural filaments offers connections to synapses and other types of nerve cells. So these are really good for overall support, and structure, right? They're kind of like the concrete that you put under the house for foundation. Without the intermediate filaments, everything would just literally fall apart. Now again, if we look at the nucleus, inside the nucleus we have the nuclear lamins, and that is present within all animal cells. And of course this offers structural support for the nuclear envelope. Okay, but what makes the intermediate filament so strong? Well, if you look at how it's created, we can see the strength immediately. So if we have one strand, and this is called the alpha helical region, this is a monomer. So this is a monomer. So again, monomer just means one. So it's just one strand. And notice that we have the N-terminus and we have the C-terminus. So these are amino acids linked together. So we have amino acids linked together. and they keep linking until we have a full alpha helix. Okay, so what if I get another monomer? So what if I have two monomers? Well, if I have two monomers, again, we're still going to have the N-terminus at one side, and we're going to have the C-terminus on the other side. But then if I get those monomers and I start twisting them together, I now have a coil, and this coil is called a dimer. This is called a dimer because dimer means two, right? Dimer means two. So if I take two monomers and I twist them together, then I get a dimer. And of course, when I do this, I have the N-terminus on one side and have the C-terminus on the other side. Now, what happens if I get two dimers and I put them right next to each other? Well, if I put them right next to each other, then I'm going to have a tetromere, because a dimer plus a dimer will give me a tetromere. Now, tetromere means 4. So 2 plus 2 is equal to 4. Dimer plus dimer is a tetromere. But notice something different. Right here we have the N-terminus, and over here we have the C-terminus. Over here we have the C-terminus, and over here we have the N-terminus. So we can say that over here, if you want to look at a simplistic image right here, we have the N-terminus on one dimer, and then we have the C-terminus on another dimer. Over here we have the C-terminus, and over here we have the N-terminus. So what does that tell you? That if we go from this way to that way, because we go from N-terminus to C-terminus when we're reading, we notice that this is more of an anti-parallel. So this is an anti-parallel fashion. Therefore, when we are creating the intermediate filaments, we go from the N-terminus to the C-terminus, and we do that anti-parallel. Okay? So they go in opposite directions. We connect them in opposite directions. Okay? So that is usually tested on exams. They'll often show you a picture that has something like the NH2 right next to another NH2, and then this C-terminus next to another C-terminus. But that's wrong. Notice these arrows. These arrows are saying, okay, we have the N-terminus going from left to right, and then here we have another arrow, meaning that we go from right to left, meaning that the N-terminus is on the right-hand side. So again, this is more of an anti-parallel fashion. So if I get 8 of these tetrameres, and I combine them going from the anti-parallel, right? I can combine them and I can make a full intermediate filament. So again, I will repeat myself. If I take 8 of these tetrameres, you can count them, 1, 2, 3, 4, 5, 6, 7, 8. If I take 8 of these tetrameres and I line them up again in an anti-parallel fashion, I can create an intermediate filament. And so we have so many coils and twists that make up one filament, and if we combine them together to make an intermediate filament, well, this is obviously very, very strong. So this is why we use intermediate filaments for strength and support, because they are created by little tiny proteins that are twisted. And if you twist something, eventually it will become tough and hard to break. And so these intermediate filaments are very tough and very difficult to break. Here we have the example inside epithelial cells. So again, this is called an epithelial cells, or I guess epidermal cells. So we have the epidermis. And that is kind of like the skin on your hand or on your chest or whatever. And notice that since we have these intermediate filaments, and if you look closely, we have these black dots. Those black dots are called the desmosomes. These are kind of like the little portals that the intermediate filaments can go through. So we have the desmosomes, and I can't really spell, so right, and that offers strength. So if I apply a pressure, then I stay intact and I am together. I'm not burst, right? But notice that we don't have the intermediate filaments on this slide or on this picture. And so if I apply a downward force, then these cells will immediately rupture and they'll be destroyed. So here, within this nice photo, we have the intermediate filaments. And notice that we are intact. However, if I don't have the intermediate filaments and I apply pressure, then the cells will become ruptured and they will be destroyed and unusable. So therefore, intermediate filaments are always used for structural support and also cell strength. But we have to ask ourselves, what stabilizes the intermediate filaments? So what stabilizes the intermediate filaments would have to be plectins. So plectins are here in green. Here is the plectin, right? So the plectin is green in this picture. And notice that the plectin is directly attached to this blue filament. And the blue filament, that is going to be the intermediate filaments. So the intermediate filaments, those are blue in this picture. Notice that the plectin is directly in contact with the intermediate filament. Now, the reason why they do this is because the plectins can link them to other intermediate filaments, and then the plectins can link the intermediate filaments with other cytoskeleton components. For instance, here in this example, we have the plectin. It is connecting to the intermediate filament, and then it is connecting this intermediate filament with the microtubule. So here the microtubule is red, and unfortunately I used red to label plectins, so we'll just say that the red is the microtubules. So what I'm saying here is that the plectins stabilize the intermediate filaments, and they also connect them to other cytoskeleton components. For instance, unfortunately this photo doesn't have it, but for instance, the plectins can connect the intermediate filaments to the microtubules and also to the microfilaments, which would be actin. So without plectins, the intermediate filaments would not be functional. We will now be talking about the microtubules. So the microtubules are a bit thicker. Here you have the microtubules, and what they do within the cell is that they do cell organization. They do cell organization. And then they also do transport. So they do intracellular transport. Okay? So again, this is going to be the microtubule. And then they also do the spindle assembly, and they function for the spindle. Okay, so within remember when you're doing cell division via mitosis, we have these little spindles that go to the sides of the cell and they shoot out these filaments. These right here are the microfilaments, and so we have the centrosome spitting out these microtubules, and eventually they will create a cleavage furrow, and this cleavage furrow will eventually split and create two cells. But this is only possible with the microtubules. So again, they make the spindle assembly. They do spindle assembly, right? Assembly. What else do they do? Well, the microtubule is responsible for the structural support of the cilia and the flagella. So they also make, so it makes the cilia and flagella. And we will go into detail about all these processes within the microtubule. However, first we have to ask ourselves, what is a microtubule made of? How is it made? Well, again, the microtubule is made from a dimer, and a dimer means two parts. So here we have the beta part, and here we have the alpha part. Now, these beta and alpha components make a heterodimer, okay, and this is called a microtubule subunit. So now each subunit will be used to create kind of like a noodle shape. So have you ever played with those little pool noodles? Well, those pool noodles kind of look like these microtubules, because microtubules themselves are hollow. So these are hollow. These are hollow tubes, and these hollow tubes are made from protofilaments. Now, the way these protofilaments are made is that these heterodimers stack on top of each other. But notice that they have an orientation, they have a direction. The beta part, the beta component, is the plus end, and the alpha part is the minus end. And the way I remembered it when I was in university is that the plus end has a beta, so we call that the bless end. And then this one is kind of like the anion. Anion has A. Anions are negative, so we have the anion, which is the alpha, right? So we have that. But notice that really, they're not really negatively charged, and they're not positively charged, okay? This is just convention, right? So scientists wanted to make this easier to learn. So they say, okay, well, what if we just treat them like they have charges? They really don't. But it's just easier to remember that the plus end has the beta. And so we go from negative, plus, negative, plus, negative, plus, negative, plus, or you can say alpha, beta, etc., whichever is to your liking. But just remember that they are not physically charged, okay? This is just sign convention. They don't have a charge. Well, how many protofilaments do you need to make a microtubule? Well, it turns out that you need 13 protofilaments, okay? So you need 13 Phineas and Ferbs to make a protofilament, okay? So 13, that's the magic number. And here we have these pictures. This is looking from the top view, okay? So looking at the pool noodle, we see that there are 13 protofilaments stacked right next to each other inside a circle. Now the diameter for the microtubule, again, would have to be 25 nanometers. And here we have kind of like a side view of what we're looking at. We can see that each protofilament makes a nice clean line going upwards, okay? So again, these are pretty cool. Just remember that the plus end is the beta and the minus end is the alpha, and that each protofilament subunit is made from the beta component and the alpha component. Within the cell, you will often have centrosomes. Now, centrosomes are kind of like little spheres that migrate to the ends of the cell, and they radiate microfilaments. But inside the centrosomes, you have two little items, and these two little items are called centrioles. So within a centrosome, you have a pair of centrioles. So what do these centrioles do? These centrioles actually have a gamma tubulin ring. So it's kind of weird, but what you have to remember is that the beta ends are radiating outwards. So the beta end expands. But inside the centrosome, we have the alpha end, and the alpha end does not expand. Rather, it is anchored. This is anchored. So alpha is A. A in anchored means alpha, meaning that we are always inside the centrosome. And really, we're attached to the gamma tubulin ring within the centrioles. So really, the alpha subunit is attached to the gamma subunit within the centrioles. Okay. So here we have the gamma tubulin ring complex, and that's in red. Notice that in red, they're really they're basically attached to the centrosome. And here we have the beta component, and here inside we have the alpha component. And so the main thing that you have to remember is that whenever we expand, whenever we are shooting out these microtubules right here, this will always be the beta end, and attached will have to be the alpha end. So this is negative and this is positive. They don't have a physical charge, it's just sign convention. The alpha ends are always within the centrosome, and really the alpha ends are attached to the gamma tubulin ring complex. That is just like an anchor for the alpha subunits. Here we have this very cute picture. So notice that we can have some beta subunits expanding, and we can also have some beta subunits shrinking. So here we have that the microtubules are dynamic. They are dynamic. Okay? So they are dynamic because they grow and they shrink rapidly. Okay? So they have a dynamic instability. So they have dynamic instability. Right? So they're always constantly growing and shrinking and growing and shrinking, but it's always the beta end that grows and shrinks. It's always the beta end that grows and shrink. Here's the alpha end, right? Here's the alpha end, or let's do this better. And this alpha end is connected to the centrosomes via the gamma subunit. So this alpha anchor is connected to the gamma subunit. And here we have this dynamic instability. However, you always have to ask yourself, how does the microtubule grow and expand and shrink? How does that happen? Well, it actually happens by using GTP energy. So really, the beta subunit, so we can write that the beta subunit is a GTPase. So it can accept GTP and it can also hydrolyze the GTP. So in this example, let's say that we are expanding. Okay, so we have a centrosome and we are constantly expanding this microtubule. The way we do this is that we take a dimer. This dimer has an empty pocket, and the empty pocket can either have GDP, diphosphate, or it can have GTP, triphosphate. Now in this example, since we're expanding, we're going to be having the guanosine triphosphate. So when this dimer has the guanosine triphosphate, it will add to itself. So we have the negative portion and we have the positive portion. So the negative portion is going to attach to the positive portion, and the positive portion will attach to a negative portion later on. So as long as we have the guanosine triphosphate, we will keep adding more dimers going onwards and onwards, and this is what you see as expansion. But if we decide that we need to shrink, then we can do hydrolysis of the guanosine triphosphate. If I do the hydrolysis of the guanosine triphosphate, I will convert it to guanosine diphosphate. Now when this happens, the structure becomes very unstable. Okay, so this structure becomes so unstable that it begins to curve. So now we have a curved protofilament, and this curve is not stable. Therefore, it starts to break apart. Now when this starts to break apart, this is what you see as shrinkage. So now this is expansion, which is GTP, and this is shrinkage, which is GDP. Okay, so that is really dependent on the hydrolysis of GTP. And here's another picture showing the expansion. So here we have the tubulin dimers, and they're bound with GTP. As long as the GTP is not hydrolyzed, then we will continue to add more dimers to the protofilaments. And eventually we will keep expansion. So this is expansion. So if I inject a drug into a cell, and this drug inhibits the process of GTP hydrolysis, that means that the GTP will always continue to be functional. And if that's the case, then this protofilament will always continue expanding. And that could be pretty bad depending on the situation. Now, this is called the GTP cap. So if GTP is always being formed and these dimers are connecting to each other and they don't hydrolyze, then we just call this the GTP cap. So we can say that the GTP cap favors microtubule growth. So this favors microtubule tubule growth. However, if I hydrolyze the GTP, then all I have is guanosine diphosphate. And remember that guanosine diphosphate within the protofilaments makes a very unstable protofilament. So here we have the protofilaments which contain the guanosine diphosphate, and they begin to peel away. So this again is called shrinkage. Right? So now you see the guanosine diphosphate tubulin is going to be released, and really you release it to the cytosol. So here we have the guanosine diphosphate tubulin. So that is the shrinking microtubule. Sometimes I need to transport certain molecules to different parts of the cell. So to do that, I need to create highways, right? So I have to get one of these items from the nucleus or whichever part, and I need to transport it to the different part of the cell. So how do I do that? Well, in order to create these highways, I need to stabilize the microtubule. And the way I do this is that I use a capping protein. So this microtubule capping protein will eventually connect. It will connect to the beta portion of the microtubule. And when this happens, the capping proteins actually prevent GTP hydrolysis. Now, if I prevent GTP hydrolysis, what does that mean? Well, it means that I will always have GTP, and that means that I will always be expanding and growing. So here we can see that this microtubule is continually expanding. It is continually growing. So this allows for cell polarization. And what that means is that the cell kind of gets a little bit bigger on one side compared to the other. So now here we have the stable microtubules, and over here, which is not connected to the capping protein, these are called unstable microtubules. And again, we do this to create intracellular transport networks. So here is your newly created highways for the cell. And here we now have the intracellular transport that is facilitated by the microtubules. So microtubules, they guide the transport of organelles, vesicles, and other macromolecules. And for instance, in this example, we have the nerve axon. So here we have this little green thing, and this green thing is the microtubule. So notice that since we created a highway, my little vesicles, or my little cars, are able to go from the nerve body all the way to the axon terminal. So let's say that these little cars contain dopamine. So this red little guy contains dopamine, and we got to transport it to another area. So therefore, we're going to be using the microtubule highway. And now my little vesicle, my little car, is able to transport its cargo from the highway to its destination. Now, there are two types of cars in this example. We have the cars going to the axon terminal, and then we have the cars going away from the axon terminal. And then we have two different names for them. So the guys who are going away from the cell body, let's call this the negative side, and let's call this the positive side, just for convention. The cells that go towards the positive side, those are called kinesins. Those are called kinesins, right? So these are called kinesins, and they go away from the body, away from cell body. Okay, but what about the negative, quote-unquote negative proteins? Well, those are called dinins, right? So those are called dinins. So we have dinins or dinines, and these go toward the cell body. Now just remember that dinins and kinesins, they don't really have charges. It's just for convention. It's just to make it easier for you to remember. So kinesins go away from the body, so they're going to go toward the axon terminals, and when they release their cargo, they're going to go back towards the cell. In that case, they're going to be called dyneins. Okay, so dyneins or dinines, they go toward the cell body. And now we will actually zoom in even more and explore the structure of kinesins and dyneins, or dyneins, whatever you call them, however you pronunciate. Here we have the kinesins and the dyneins, and notice that they're using the microtubule as a highway to transport their cargo. Right, so these motor proteins move along the microtubules, but they do it in opposite directions, and they use their heads to connect to the microtubules. So their heads connect to the microtubules. Heads on microtubules. So again, the kinesin go towards the plus end, and the dyneins go towards the negative end. And how do they move? Well, they move using ATP hydrolysis. So they use ATP hydrolysis. Okay, so you can kind of imagine a person walking down the street. Every time this head attaches to this microtubule, ATP is hydrolyzed, because whenever you cut off a phosphate bond from ATP, it releases a lot of energy, and that energy is going to be used to power the motor proteins along the microtubule. And here we see the microtubules, so not the microtubules, but the motor proteins. The motor proteins going towards the plus end, away from the cell, those are called kinesins, and the motor proteins going towards the cell are called dyneins. Now, notice that sometimes we have different cargoes, so depending on where you're at, the entry and the exits of the motor proteins will have different cargoes. So, for instance, let's say that this is dopamine, and this is serotonin. And what if this is acetylcholine? What I'm saying here is that these motor proteins are constantly transporting something. Sometimes, if you have a drug that inhibits the formation of motor proteins, you won't have the dopamine being transported into the nerve cells. And if that happens, then most likely you're not going to have proper function. So if you don't have serotonin, you're not going to be happy. You could have depression. So really, depression could come about from having an inoperative motor protein. So now I will show you a video about the motor proteins and how they operate.
Motor protein kinesin is a dimer with two identical motor heads. Each head consists of a catalytic core and a neck linker. In the cell, kinesins pull organelles along microtubule tracts. The organelle attaches to the other end of the long coiled coil that holds the two motor heads together. The organelle is not shown here. In solution, both kinesin heads contain tightly bound ADP and move randomly, driven by Brownian motion. When one of the two kinesin heads encounters a microtubule, it binds tightly. Microtubule binding causes ADP to be released from the attached head. ATP then rapidly enters the empty nucleotide binding site. This nucleotide exchange triggers the neck linker to zipper onto the catalytic core. This action throws the second head forward and brings it near the next binding site on the microtubule. The attached trailing head hydrolyzes the ATP and releases phosphate. As the neck linker unzippers from the trailing head, the leading head exchanges its nucleotide and zippers its neck linker onto the catalytic core, and the cycle repeats. In this way, kinesin dimers move processively, step by step, along the microtubule.
The microtubules that radiate from the basal body, so we have the basal body of the cell, that would be this right here, right? So this region. The microtubules that radiate from the basal body, they are able to support the cilia and the flagella. So basal body microtubules support the cilia and what else? The flagella. Okay, so here we have some examples right here. So this is a sperm, right? So this is the spermatozoa, sperm. And notice that we have the head of the sperm, and that is attached to the flagella. So the flagella is actually made from the microtubules, and the flagella kind of flap around in the liquid or the fluid, and that propels the sperm to travel in the vaginal canal. So this portion here is made up from microtubules. In this example, we have the cilia. So the cilia is different from the flagella. They're both made from microtubules, but the cilia is anchored to the plasma membrane. So the cilia is just to kind of propel items to different areas. So let's say that we have a hot dog, right? Or some tacos or something. So this taco particle is going to come into contact with the cilia, and the cilia would just kind of push the fluid to that section. So the fluid is going to be propelled by the cilia, and it will carry the items to different parts of the environment. So here we have the cilia, and they look like little fibers radiating out of the cell. And again, these cilias, they just push fluid around. So the difference between flagella and cilia is that the cilia moves fluid from the it moves fluid over the cell surface, but the flagella is for transportation and locomotion. So this is locomotion, and this one is just for moving fluids. Now, microtubules, since they support flagella and the cilia and the flagella actually have the same structure. If you were to cut a flagella in half and look at the flagella, you will see this. You would actually see this. Now, these two circles that you see here, these are dimers. But what are they made of? Well, this on the left, this is made from microtubule, and this one on the right is another microtubule. So if we call this one microtubule A and this one microtubule B, notice that there are a pair of microtubules. Within the cilia and the flagella, there are 9 pairs in the outer ring of microtubules. Inside the inner ring, there is 1 pair of microtubules. So we call this the 9 plus 2 conformation or arrangement. So let's write arrangement. Right. So we have 9 pairs of microtubules on the outside ring, and we only have 1 pair of microtubules on the inside ring. So this is molecule or a microtubule A, and this one is microtubule B. So that is 1 pair. So it's a bit weird why they say 2, but just know that the inner ring just has 2 microtubules, microtubule A and microtubule B. So that makes 2. Now they are connected, the outer pairs are connected via a protein called the Nexin protein. And this Nexin protein is kind of like super glue, and it allows the microtubules to just stay constrained and stabilized. Right? So the Nexin is very important, and we will talk about Nexin in the next slide. Haha. If we zoom in to the microtubule structure, we will notice that there are kind of these motor proteins between the microtubules. So let's say that this is microtubule A and this is microtubule B, and together they make a dimer, which is a pair. Now between A and B, you will find dyneins or dyneins. So here, this is that, dyneins. Now these are they are responsible for the sliding filament model. So between the microtubules A and B, whenever we need to move, the dyneins will actually shift. They will do conformational changes, and this produces the microtubule sliding. And within the flagella specifically, in order to do movement or locomotion, the dyneins are going to be moving around, and they actually cause a bend, right? So this is actually curvature, so curvature. Now, the reason why this curvature is allowed to happen is because we have the linking proteins. Now remember, the linking proteins are called nexins. Without these linking proteins, you wouldn't have the ability to move, right? So if dynein shifted, if it did a conformational change, then most likely this structure would be unstable and the flagella would not be able to operate. So if you were to inject a toxin that dissolved the linking proteins, then you can predict that the sperm, in this example, would not be able to move, because the linking proteins is what allows the microtubules to do a curvature, right? So in a normal flagellum, dynein causes microtubule bending, and it is these linking proteins that stabilizes the microtubules. Without these linking proteins, these bending microtubules would break apart, and they would not be stable. We will now be talking about microfilaments. So remember that microfilaments are filaments, are filaments made from actin. Within the body, there are 4 categories. So there is the stable category. There is another stable category, and then these are the temporary categories. So I guess not really 4 categories, but rather 2. It's very difficult to talk and write, but yeah, it's whatever. So within these categories, we have different items. So for instance, this right here, what would you think this is? This is a microvilli. This is a contractile bundle. This right here is the protrusions. These are protrusions. And this is for cell movement. You could kind of think of this as pseudopodia. And right here, which looks like kind of like this belt, this is actually a cleavage furrow, and you often see that within cytokinetic, cytokinetic contractile, cytokinetic contractile ring. And so the microvilli or microvilli, this is needed for absorption of nutrients. So you often find these within the epithelial cells of the intestines and the gut. So this is for absorption of nutrients. This is food processing. It's food processing. This contractile bundle, it kind of allows the cell to maintain a shape. And so we have this predefined shape that is actually stabilized by these microfilaments. So this is a shape. This is for movement, and we'll talk about that later on. So this is movement. And this is for cell division. Microfilaments are made from actin. So an actin filament, which is a microfilament, is obviously made from actin. And they're actually composed of globular actin proteins. Okay, so the type of actin that we're using is a globular. This is a globular actin. Now, similar to the microtubules, in which that had polarity, the actin monomer also has a minus end and a plus end. Now you're probably wondering, well, if we're working with a monomer, which one is negative and which one is positive? Again, positive and negatives, that's just convention. There is no physical charge on this actin monomer. However, you have to remember that for the actin monomer, it is most likely added to the plus end. So this is always added to the plus end. And so if on an exam you see a picture where these actins are being chained together going this way, then instinctively you can say, okay, since the actin is being added going from left to right, this must be the positive end, and this must be the negative end. If instead you see the actins being added going from right to left, depending on your professor, how they draw it out, then you can say, okay, in this example, the actin was added going from right to left, so that must mean that the left side is a positive side, and that the right side is a negative side. Now, when we are creating the actin filament, we actually twist the fiber. And we do this twisting motion because that offers a greater strength towards the overall filament. When we are creating an actin filament, we are working with ATP. Therefore, unlike microtubules, the microfilaments are actually ATPases. So actin is an ATPase. Remember that microtubules, which works with alpha and beta subunits, that's a GTPase. But the actin is the ATPase, and is very similar to the microtubule, in which if I keep on using ATP, then I will continue to expand. So notice that when we are expanding, ignore everything else right now, whenever we are expanding, we are bound to ATP, and I will continue to grow. However, if I begin to break down the ATP and work with ADP, if I do a hydrolysis of ATP, then over here I will have the unbound section, right? So this is the shrinkage. And that is bound with ADP. So let's kind of recap. If I want to grow the actin filament, I will be bound to the ATP and I will continue to grow. However, if I take away the ATP and use ADP, adenosine diphosphate, then this becomes unstable and it will actually shrink. So in both cases, within the microtubule and the microfilament, if you hydrolyze, if you take away phosphate group from either the GTP or the ATP, you will have an unstable filament, and therefore shrinkage will occur. Within the cell, actin is in little bit of quantities. So actin, if you want to look at statistics, only 5% of the protein in all our bodies is actin, right? So 5% of proteins in the cells is actin. And half of this is actually assembled into microfilaments. So these are microfilaments. And the other half is created into monomers. So this is monomers where? Well, they're monomers inside the cytosol. So the cytosol. Now, the monomer can actually bind to the monomer sequestering proteins. So what is a monomer sequestering protein? Monomer sequestering, well, first of all, sequestering means to grab. So this just means monomer grabbing proteins. And they just kind of like hold on to it for later reactions. So if I need actin for whatever reason, these proteins can transport the monomers and they could do different reactions. Or if I need them to do something else or to be transferred somewhere else, they can be transferred. But sometimes the actin monomers bind to the nucleating protein. And the nucleating protein promotes actin polymerization. So this promotes actin polymerization. Now you're probably wondering, well, polymerization, that's a big word. I'm kind of scared. What does that mean? This just means growth. So in the last slide, when we saw that actin monomer was growing, that was called polymerization. And these nucleating proteins, they stabilize the actin monomers. And when they stabilize the actin monomers, they are attached to ATP. So the nucleating proteins prevent the ATP from being hydrolyzed. And if I prevent the ATP from being hydrolyzed, that means that I will continue to expand. And this expansion is called polymerization. Now you will later on see that the nucleating protein is useful and is actually important for the cell to move around in its environment. Cell movement is composed of about 3 steps. So the first step will have to be the polymerization of actin on the right-hand side in this picture. So whenever the actin is polymerized, it will continue to grow. So notice that this section kind of grew longer. Now when this occurs, we have to have contraction of the back portion, and this back portion is the actin shrinking. Now later we will go into each step in more detail. In the first step that you saw for cell movement, you saw that the actin was polymerizing at the leading edge. Now this actin polymerization, that involves 2 structures within the cell, and that is the lamellipodium and the filipodium. Now the difference between the lamellipodium and the filipodium is that the lamellipodium is called the branched. This is the branched actin network. And the filipodium, that is called the unbranched actin network. So, that was bad. When these 2 structures, the lamellipodium and the filipodium, work together, then they actually make the cell move quickly. Therefore, if the cell only has one lamellipodium working and the filipodium is not operating, then the cell would not be able to move around. Therefore, you need both the lamellipodium and the filipodium in order for the cell to move, in order for this actin to be able to polymerize. The way that the cell moves is that whenever actin is polymerizing, it will polymerize towards the leading end of the cell. How does that happen? Well, it happens, so actin polymerization occurs with the ARP complex. Now the ARP complex stands for actin-related protein. So that is actin-related protein. And this protein complex is going to stabilize the actin monomers. And when it stabilizes the actin monomers, the ATP cannot be hydrolyzed. And when the ATP cannot be hydrolyzed, the actin monomer begins to expand. Now, if you didn't have these capping proteins, which we'll talk about in a few seconds, then you wouldn't be able to move in a controlled manner. Why? Well, these capping proteins, they attach themselves to the expanding end of the actin monomer, and they prevent the actin monomer from expanding even more. Now they do this to certain monomers in order to create a direction. So if I did not have these capping proteins, then I would have expansion everywhere, and I wouldn't have a localized protrusion. So what I'm saying here is that the capping protein is able to direct the actin polymerization towards the leading edge of the cell, and that gives it direction, and that allows us to go forward. If I did not have these capping proteins, then I would just be going everywhere, right? I would have a cell expansion and I wouldn't have any directions. So you need both the ARP complex and the capping proteins in order to move the lamellipodium. So again, this actin polymerization in this example occurs at the lamellipodium, right? So this is the lamelli podium. That's usually the section of the leading edge. In step 2, the actin monomers must attach themselves to the integrins in the plasma membrane. So these integrins are called transmembrane proteins. These are transmembrane proteins that are attached to both the external environment and the internal environment. And specifically, these integrins, they are connected to the extracellular proteins and some polysaccharides. So they're always connected to polysaccharides, collagen, fibronectin, etc. Right? And that just offers them more support and more structure for their bodies, okay? So just remember that integrins are both connected to the extracellular matrix and the cytoskeleton. Now, the integrins, they have two sets of hands. One hand is connected to the extracellular matrix, for instance, let's say a polysaccharide, and the other set of hands is connected to the actin filament. Now, these integrins are going to offer support for the actin filament. So this offers like a stabilization, stabilization for actin. Right. So it just offers actin an extra grip onto the plasma membrane, and that is step 2. In step 3, we finally have contraction. In order to contract, the actin must associate with myosin, and when it associates with myosin, it forms a contractile structure. In this case, the simplest myosin is myosin 1. This is the simplest myosin. And what this does is that the myosin head is going to directly attach itself to the actin filament. And the tail, this portion, is the portion that connects to the cargo. Now, you have to remember that in this case, myosin is going to be going towards the plus end. So in this case, you need to remember that myosin goes to the plus end. And what is the plus end of the actin filament? That is the growing section. So this is the growing section of actin. And you have to remember that myosin 1 is involved in vesicle movement. Okay. And here in this lower picture, this is showing you that it can also bind to the actin filament within the cortex. Now, why would it want to bind with the actin filament within the cortex? You have to remember that a couple of lectures ago, I mentioned that the cortex has actin, and this actin stabilizes the cell shape. Now, the reason why myosin would want to bind with the actin filament within the cell cortex is to establish a new shape. So this makes a new cell shape. And so if the shape of the cell is, if the cell is constantly changing shape, and we want to kind of make that shape secure, and we want to stay in that shape, then we would use myosin 1. Okay? And again, the reason why this arrow is showing you it's going for the left side is because the myosin, they pull the actin going towards the positive end. So get a piece of string or something, or a pencil, and if you have your pencil in your right hand, your pencil is the actin filament, and your hand is the myosin head. So the myosin head is going to grab the negative end, and it's going to pull. And when it pulls towards the left, notice that your right hand is going to go towards the positive end. And so what I'm saying here is that your hand, when it pulls going towards the left, the pencil goes towards the left, but your hand, the myosin, goes towards the right. And I recommend that you do that on your own. And that's how you actually understand the concept of myosin and actin contractile polymerization. Okay, so just use a pencil and do that example. Well, now we're dealing with something else. This is still microfilaments. But you're probably wondering, okay, we can have cell polymerization, we can have new shapes, and all these great things, but how does the cell know what to do. Remember when I was talking to you about cell signaling? Well, cell signaling is to blame, or is to, we're going to blame it. So we're going to blame extracellular signal to different networks, okay? So really, these cell signals, they converge. That means that they connect to a family of row GTP binding proteins. And if you need another word for this family, they're really a family of GTPases. And so when this family receives a signal from the extracellular space, they have three family members, okay? So first of all, we have the row family member. And the row family member, that promotes contractile bundles. The other family member would have to be the rack. So the rack family member, which is also another GTPase, that promotes the Lamellipodia, which is cell movement. And the third family member is CDC42. And CDC42 promotes Filopodia. Which is also necessary for cell movement. Here we have in picture A cell that does not have any signals. And with no signal, the actin is relatively normal. It's not moving around. But if we get a signal, and that signal talks to the row family member, then the actin within the same cell will start to go crazy, and it will actually create a contractile bundle, and that kind of strengthens the cell membrane of the cell. If the signal talks to the rack family member, then we will actually expand. So notice that this cell looks a little bit smaller, but when we have the signal that goes to the rack, the cell expands, right? So it's getting ready to move around. And here we have the same cell that finally receives a signal for the CDC42. And when this occurs, the Filopodia expands to the external environment. Within the body, there are multiple muscle contractions occurring right now as we speak. And the way that these muscle contractions occur is through the usage of Myosin 2. Myosin 2 will interact with the actin filaments and create a contraction. Here we have one molecule of Myosin 2. If we take one Myosin 2 and connect it to another Myosin 2, and then we twist it, we create a dimer. Now what if we take a bunch of these guys and start twisting them on each other? If we do that, then we have the Myosin Two Filament. Now this is very similar to the creation of the intermediate filament. So if you need to be reminded of how this is created, just watch how intermediate filaments are created. It is the same process. Within this Myosin 2 filament, you will notice that there is a bare region. This bare region is the part of the filament that does not have a myosin head. Okay, that will be important later on. Now also notice that whenever the myosin filaments, here's filament 1 and here is filament 2, whenever these filaments are attached to another, they will interact with the actin monomer, with the actin filament. And when this happens, they are going to contract to the opposite portions. Okay, so this portion right here is going to go to the positive end, and this portion right here is going to go to the other positive end. Notice that these myosin heads are separating from each other. One is going towards the left, and one is going towards the right. This causes a contraction. Okay, it's kind of like tying a knot and then pulling the strings apart from each other. When you do this, the knot will contract and become tighter. Within the body, there are muscle cells, and these muscle cells are comprised of myofibrils. So these right here are called myofibrils. These are called myofibrils. And these myofibrils, they make up the skeletal muscle cell. Okay? So the myofibrils themselves are made up of sarcomeres. So let's kind of recap. The myofibrils make up the muscle cell, but the myofibrils themselves are made up of sarcomeres. This is the basic unit of a muscle cell, right? And now these sarcomeres are actually the contractile units of muscles, right? So the sarcomeres they themselves contract, and whenever they contract, thousands of these sarcomeres will kind of squeeze together, and they will cause thousands of myofibrils to squeeze together, and this causes this causes a muscle contraction. And now we will zoom in on this process of how myosin and actin cross with each other to cause the sarcomere to contract. This is the sarcomere zoomed in. Now before I begin this portion of the lecture, I want to break down the sections of the sarcomere. Here in the bare region, if we were to make a line, this would be called the M line. And this M line is called M because it is for the middle. Here in this section, we have the Z discs. So on the left portion and on the right portion of the sarcomere, these are the Z discs, and this causes boundaries. So a sarcomere is defined from one Z disc to the other Z disc. Okay, this portion right here, let's write that better. So this portion right here, where it's only actin, well, that is called the I band. So this is the I band. Now the I band only has actin. This is only actin. And if I were to incorporate this portion, where it's like a combination, right? So this combination, this is actually called the A band. This is the A band. So A band, you can remember it from A. A means to alternate. So A band has the actin and the myosin. Similarly, if I were to look at this band right here, where it's only the myosin, well, that is called the H zone. That is called the H zone. And the H zone only has myosin. So to recap, the sarcomere is defined by the boundaries of the Z discs. The middle portion of the sarcomere is called the M line. The portion of only actin is called the I band. The portion of the sarcomere that combines the actin and the myosin, that is called the A band, and that is called alternating. The portion of the sarcomere that only has the myosin is called the H zone, and that only has the myosin. Here we have, in order to contract, the Myosin Two heads will associate with the actin filaments. Now the Myosin Two heads will go towards the plus ends. They have the plus ends, okay? So the myosin heads will connect with the actin filaments and go towards the plus end. Here in the bottom picture, we have the contraction. So notice that the sarcomere kind of squeezed together, okay? So it's squeezed together. However, you have to remember and you have to know that the sarcomere does not contract, okay? The sarcomere does not contract. Rather, we just kind of pull the sarcomeres closer together, okay? So the myosin heads are going to grab the actin filaments and they're going to pull the actin filaments closer. However, everything is the same. If we have, let's just say, for example, 50 centimeters of actin, we will still have 50 centimeters of actin. We will not have 49 centimeters 48 centimeters or even 53 centimeters, it will always be a fixed length. Okay, we are just kind of pulling the sarcomeres closer together. Okay, so again, the sarcomere does not lose length, it does not gain length, it just kind of contracts, it just gets a little closer together. Okay, everything is the same length, we are just pulling the sarcomeres closer together. However, we have to ask ourselves, how does the myosin connect to the actin filament? What happens here is that at the start of the power stroke, this is the power stroke cycle, so let's say stroke cycle, I can't spell, is that at first the myosin is bound to the actin filament. However, when ATP binds itself to the myosin head dislocates from the actin filament. So ATP binds to myosin. And when it binds to myosin, the myosin releases actin. Now, this actin molecule right here will actually be hydrolyzed. And when it is hydrolyzed, it will shift the myosin head into a new conformation. So previously, the myosin head was in this conformation and it had the ATP. But when the ATP hydrolyzes, it releases energy. And that energy is going to be used to shift the myosin head forward. And when it shifts forward, we now have this new conformation. Afterwards, the inorganic phosphate ion will be removed from the myosin head. And when this happens, the ADP is released and the myosin head kind of moves the actin filament. So to recap, the ATP is hydrolyzed. And when this happens, it releases energy. And this energy shifts myosin head forward. And when it shifts forward, it will come into contact with the actin filament once again. Now the ADP and the inorganic phosphate ion will leave. And when this leaves, the myosin head will move the actin filament to the left side. Okay? So now actin filament will shift. And then once again, the ATP could come in here and it can bind to the actin, to the myosin filament once again, and it can cause a conformational change. And that is the power stroke cycle. Within muscle cells, we have the sarcoplasmic reticulum. And the sarcoplasmic reticulum will be highlighted right here. So it's kind of like this inner cell right here. Now the sarcoplasmic reticulum has calcium ions. And remember from other biology courses, calcium is needed for all muscle types. Right? It is needed for contractions. Right? And so I need to find a way to get this calcium outside of the sarcoplasmic reticulum. The way I do this is that an action potential will propagate along the muscles and release voltage-gated calcium channels within the sarcoplasmic reticulum. And when the sarcoplasmic reticulum opens up, it releases the calcium, and the calcium can bind to the muscle cells and cause contractions. However, I need to get this action potential within the muscle cell. So how do I get these action potentials into the muscle cell? Turns out that within the muscle cells, we have these little loops. And these little loops right here, these are called transverse tubules. And these transverse tubules allow the action potential from other cells to enter the cell, right? So the action potential, let's say from the brain, is going to be propagated along these transverse tubules, and they go into the myofibril, and from the myofibril, the action potential can activate the sarcoplasmic reticulum, and when the action potential activates the sarcoplasmic reticulum, it releases the calcium, and now when the calcium is released from the sarcoplasmic reticulum that was activated by the action potential, it can actually bind to the muscle cells, and that can cause a contraction. And so really, transverse tubules are needed. They are needed to transfer action potentials into myofibrils. Without these transverse tubules, the action potentials would have a longer time to get to the myofibrils, or they could not even reach the myofibril in the first place. And so therefore you would have a muscle paralysis, you wouldn't have muscle contraction. So therefore you need transverse tubules in order to transfer the action potentials into the myofibril. Here we have the transverse tubules. If I create an action potential, the action potential will be carried by the transverse tubules. And when this action potential is in contact with a voltage-gated calcium channel, the calcium channel will become released. And since there is a large amount of calcium within the sarcoplasmic reticulum, it will be released into the cytoplasm. So to recapitulate, the action potential is carried by the transverse tubules within the muscle cells. When the action potential is in contact with the voltage-gated calcium release channel, the calcium channel will open, and since there is a high concentration of calcium ions within the sarcoplasmic reticulum, the calcium ions will go down the concentration gradient into the cytoplasm, and now we have free calcium ions within the myofibril. And since there is a free calcium ion, that will cause muscle contraction. You're probably wondering what a sarcoplasmic reticulum is. Well, that is a modified endoplasmic reticulum. So really, it is just an endoplasmic reticulum that has been changed to have a lot of calcium ions in it. And these calcium ions are necessary for the muscle contraction. But let's see why we need these calcium ions. When we are resting and we do not have muscle contractions, something is covering the actin, right? So what is covering the actin? It turns out that the tropomyosin covers the actin. So this covers the actin. And whenever the tropomyosin covers the actin, well, now my myosin head, let's say over here, my myosin head cannot come in contact with the actin. Why? Well, the tropomyosin, which means above the myosin, is blocking the connection points between the myosin and the actin head. So the problem is, how do I get rid of this tropomyosin? How do I move it? Turns out that if you have calcium, the calcium will bind to the troponin complex. Therefore, calcium ions bind to troponin. And when this troponin complex is activated by calcium, it will pull the tropomyosin away from the actin. And when it pulls the tropomyosin away from the actin, it exposes the actin. And when the actin is exposed, the myosin heads can connect to the actin and cause a muscle contraction. Therefore, we need calcium ions to activate the troponin complex. An easy way to remember is that troponin, it has an N. So troponin, calcium, in troponin. Calcium in troponin. Okay? Calcium goes into troponin. tropomyosin is moved by troponin. Now let's put it all together. When we need to make a muscle contraction, calcium needs to exit the sarcoplasmic reticulum. This occurs when the action potential is transported via the transverse tubules. When the action potential is exposed to the voltage-gated calcium channels, they will open, and now calcium is free to move around the cytoplasm. The cytoplasm is in contact with the actin monomer. Now the calcium ions will bind to the troponin complex, and the troponin complex will move the tropomyosin. When the tropomyosin uncovers the actin site, so the myosin binding site is initially it is covered by the tropomyosin, but whenever calcium is present, the troponin will move the tropomyosin and expose this myosin binding site. Therefore, the myosin can now bind to the actin filament. So here's my poorly drawn myosin head that I kind of drew in a rush. So there you go, and that is a muscle contraction. So hopefully now you understand how muscle contractions occur, the different parts of the cytoskeleton, how microfilaments build and expand and shrink, how microtubules build and shrink. So I hope that you understand everything that was said in this video and that you do well on your exams. And remember that I love you so much and that you are amazing. So please have a great day and be safe. And thank you for being with me.